It's tough to start talking after here. Jerome's talk really was very motivating, and I will tell you about my experience that I will start later on in life since I'm way older than all of you here. So I will start talking about before the time you were born, but still when I was starting my career. And I decided to pick this title because first I was not sure what I will be talking about, but of course when Rita's office called for inviting me to give a talk, I say yes without even looking at what I was supposed to talk about. And there I came. So thinking about how I got here, I think there is a lot of chance, just life circumstances, and but I also think that helping and be prepared is part of what let us be where we are. So I will tell you a little bit about my life path, science at least, a little bit about neuroimaging, and I don't want to get into details because otherwise we will be here for days to end. But I will point you to some places within campus that you can get more information if you are interested in. I will mention a couple of studies to show you the different type of things we can do and aspects with the same type of background and how you can go to different fields. And then just wrap up with some thoughts, and we'll see how the time goes and whether we spend more time or less. So I'm also showing you some maps. I come from Buenos Aires, Argentina. It's a big city, several million people. If you count the surroundings, it's about 11 million, probably more now. However, when I finished high school, and I'm talking 85, so long time ago, I was interested, as Jerome was saying as well, in medicine as a way to understand the human body, but also in engineering. There are a lot of tools that can be brought up to improve the quality of life, being to sustain life, to study diseases. And so I was looking for something that could be a combination of both. And I was lucky enough that the program was created in my country about a few months, or I guess a year before my time to go to college. So that was my first move was from Buenos Aires to Entre Rios. That it's kind of the opposite of what most people do. Most people come from the small towns and go to the big cities, at least in Argentina, where it's not that common to move out for college. However, I moved from the big town to the small city. Oh, there we are. To a small city in the Mesopotamia part of Argentina. So was there for a few years. The program is 6 years, where we see a lot of different things. Here I think it will be considered a mix of biology, engineering, computer science, and so forth. And within that program, I have the opportunity to work on a little bit of research, but not much. The university was fairly small. The library that we have was probably the size of the tables here, so the resources were limited. The career was new, so we didn't have many books to study. Mainly things were created as we were going. But I had the chance to, since I was in the first class, to help in the teaching to the classes coming behind us. So it was a great experience to be involved in the career. And we had at the end of the 6-year program is concluded with a dissertation, and for that I work on functional electrical simulation. So that is, we were building everything from scratch, setting up electronic circuits, doing coils and putting electrodes and put electrons on the hands of people and make them twitch. And the way that and so part of my work was programming a multiprocessor that was done in X. So it was no technology as you can have today. So your cell phone can do 100 times more than the computers we were using at the time. And I want to make a point of that because a lot of the things that we can do now in neuroscience, in medicine, are supported by the advances in technology that we have had. So while many times there is this dichotomy of people thinking that medicine is the strong, and I'm not taken out of there. I want to strengthen to those of you that are interested in technology and science from an engineering, that those are very, very valuable routes to take because will provide tools that are needed for the rest of the field as well. So going back to the electrical stimulation, after setting up the program and graduation, I did my first work at a rehabilitation institution where we could try these stimulators on patients, on hemiplegic patients. And what was very interesting is that we could see that even though we were training them or trying these devices for a little bit of time, these patients have, and this is anecdotal, right, it's not a full study and documented, but these patients seems to show a better improvement than the ones that were doing their traditional rehabilitation. And aside of that, I have the opportunity to participate in a week training on MRI, and the new thing at the time in the mid-90s, early 90s, was functional MRI. Functional MRI started in 1992, and I'm talking about in 1994. So it was very beginning field, and seems to be a way to explore the brain. That sounds really, really interesting to try to understand what was going on in the brain of these patients that were being stimulated and recovering better. So from life circumstances, I did the longest trip. That is from Buenos Aires. I landed in New Haven, Connecticut. And I find out they have magnets there. It was a big university, Yale. And I was very lucky to have the opportunity to meet Dr. Gore, that just happened to be in campus today in another of the educational symposiums, NATER. And so I make an appointment with him for, I think it was a Tuesday for Thursday. And in between, it just happened that they decided that they needed to hire someone to run the biomedical engineering lab. So I guess that was my day of luck because I already have my appointment with him. And there was my first job, and I ended up moving to the US 20 years ago. So yeah, so I went back to work on the lab and setting up the classes. MRI had to wait for a little bit for me. I could assist to the seminars and classes, but I was not running experiments then. And once the program was established, I went to grad school. So finally I can start working on MRI. I did work on multimodal studies of brain activation. I'll tell you a little bit of the studies, but mainly it's a combination of different modalities and see how we can apply the different technologies to understand the brain. Most of the things I consider myself very fortunate on the places and times that I've been. However, I love biomedical engineering, and the official department was created the day after I graduated. But, well, at least it's there. So after that, I did a little bit more of moving. I wanted to move out of New Haven, go to a different city, go to a bigger city. And the lab that I was working moved from Yale to Vanderbilt. So also I was very lucky to have the opportunity to be in great institutions that definitely is, it's always a help. So I went for a little bit to Nashville to complete some of the work that I was doing. And as I said, I wanted to come to a bigger city. DC was at the top of my list. And by the time, this is 2003 now, there was an opening in Dr. John's group here at NINDS. And he was looking for someone to do EEG fMRI. And I've been trying to do these simultaneous studies. Equipment at the time was not that great, but he had equipment and needed someone that knew the techniques. So it was a good match, and there I came, started working here in February 2004. So I've been here for a long, long time now. I will tell you a little bit more about these studies that I did there on resting state fMRI and sleep. And after my postdoc, I needed to look for a more permanent job, and decided I would do some neurodegenerative disorders, and in particular I was interested in Parkinson's disease. And so I went upstairs to talk to Dr. Hallett about some possibility to join their clinics and get a little bit more familiarized with the disorders, so I could start thinking of grants applications and try to get a transition to the real world. But again, I was lucky, and he needed a staff scientist, so I ended up staying there for the last 8 years. And I will tell you some about the studies here. So I've been working on different things over the years, but the driving force has been to understand physiology or behavior in the normal conditions and understand pathophysiology. I think physiology and pathophysiology are one of the most interesting things in the human body, and try to understand how the body works, how the systems are compensated, and how we can interfere on them to improve quality of life. It's a big challenge. And neuroimage offers us a window into the brain, into the systems. And remember, I told you I was interested in medicine, engineering, and neuroimaging is a perfect marriage of both. So when I talk about neuroimaging, I'm talking about looking into the brain, and we can look at different aspects of the brain. We can look at changes that happen in the hemodynamic and metabolic responses, and also there are electrical changes. They happen at different time scales. And so to understand the brain and use the techniques, we also need to understand what each of these methods and techniques are informing us about the brain. And then we can try to study the brain. As I said before, I'm not going to get into the details on the MRI because that will be very long, but I will suggest, hope many of you are taking this summer course that we are offering every year on neuroimaging. And these classes are open, are posted on the web. We have now, I think we're in the 3rd or 4th year of that, and all the lectures are freely available. So if you're interested in neuroimage, please go and look at the classes. Are given all by faculty here at NIH. So besides having all the technologies for studying the brain and understanding, we have the other side, right? The brain itself. That is very complex. Most of you probably have had some biology classes, and you know that you can look at different levels. You can look at the whole brain, the cortex, or you can go to neurotransmitters, you can look to synapses, you can look within a brain structure to the somatotopic organization. So you can look at different paths and different ways in the brain, and each of them will give you different information. So it's a fairly complex system to study. So depending on what you want to look into in the brain, you can find yourself in front of a different modalities of neuroimaging that can help, together with the behavioral, the clinical information, try to sort out how a system works or what is wrong in a particular disorder. And what is the best way to look at these things? It depends, right? It depends on the questions that you have. Otherwise, there is no unique technique. So one thing that is important if you're trying to study a system is don't think that because you know a technique, that's the only technique that will work to answer your question. Just think that different technology gives you different information, and different methods will give you different information, and the challenge is to put all these together. So I'm going to briefly mention two sets of studies, one that I did during my time as a postdoc here at NIH, and one of the recent projects that I've been working on. And they are quite different, both in the modalities and in the type of questions that we have, but I still have a lot of challenges and commonalities if you look at it in certain ways. So the first, I said I will talk about functional connectivity. And even though I say I'm not going to talk about methods, I do need to tell you what I'm referring to. So when we look at the brain, let me see if I can get this. If I look at the activity in this yellow point, over time I will have some fluctuations. That's it's called the blood oxygenation level dependent signal. That is how much, in a simple way, how much oxygen to the oxygen hemoglobin is in your brain that will produce different alteration in the magnetic field that would produce a signal that you can measure, and this change over time. So then we can look at the rest of the brain and see how the signal in one area relates to the other, and we could create a correlation coefficient the same way that you will do with two sets of numbers. When we get the data out of the brain, we are working with numbers. It's signal processing and image processing. It stops being a brain and biology, right? Signal processing is signal processing in whatever field, but you need to always keep the idea of what is that you're looking at, and this is what drives our question, right? This functional connectivity is very interesting. As I mentioned, if I look at two voxels, they could be correlated signals. And this top panel shows 3 different measures on the same subject on 3 different days. And the figures are quite similar. That means that these patterns of correlation show in the different days in the same way. So the areas are not next to each other, rather different vasculature, different controls, and yet they work together, or at least its fluctuations go together. What is also interesting is that now, if I do the same thing on different subjects, the yellow here means that 7 out of the subjects, I think there were 7 or 8 subjects, have the same areas fluctuate in the same way. So there is something very systematic and intrinsic about this, and sometimes we refer to this as intrinsic connectivity because it's something that we see pretty much in every condition. When it hasn't been seen, is in a deep coma state, but pretty much is seen in all conditions and everywhere. So we wanted to know what are these fluctuations, whether they are there all the time or can we modulate it, because people started using these fluctuations to try to understand disease. And so we wanted to know in a controlled way within subjects whether you can manipulate that. How were the variability that you can find in these signals? And so we choose sleep as a model because it's something that we can control. It's a continuum. hopefully, or at some point in the day, you were conscious and fully awake. After lunch and with the lights off, you may be drowsy. Hopefully, we're not getting into deep lights or deep sleep, but someone may be around here. Hopefully, I keep you awake. So we know that with this comes electrophysiological changes and behavioral changes, and we wanted to know whether the bold signal, this electrophysiological signal, was changing as well. And I'm not going to get into a lot of details because I want to get to the other study as well, but this is a time course as I showed you before, and I don't need to tell you much for you to distinguish that this part of the curve is different than this. It's pretty obvious that something is different in the bold signal. This is a time course from the visual cortex, and if you've seen the electrophysiology, also you'll see this line at 10 hertz, that is the wave that you will have when you close your eyes and you get kind of drowsy. This is the alpha wave, and this is what you have in that condition. However, when you fall asleep, the frequencies go down, the brain waves slow down, and so you can see a very distinctive pattern both in the EEG, I mean, this is a time frequency decomposition of the EEG, as you can see in the bold signal. So this is good news. The amplitude of these signals are related with your state. However, in this first study, and this, just let me add, this is the case in the sensory areas. However, we want to see whether the default mode network, that is the group of regions that are thought to be more of an introspective set of regions. So when you're doing nothing, these areas light up. And when you engage yourself in other tasks, all these areas of what is called the default mode network kind of quiet down. That's how they were defined in a set of studies. So we wanted to know whether this sleep also is modifying this connectivity. And the first study that we did on light sleep didn't show that, so it was a little bit disappointed. It was so the field was, by one hand, saying that these networks are very stable. By other hand, we wanted to study disease. So we wanted to figure out why this is not changing, and we decided to go to a next step that was looking in deep sleep. This was very involving. We had to be here overnight, scan the subjects at 2 in the morning. There was a time that were more pressure, but finally it was very rewarding because we were able to find these differences where, indeed, when you fall asleep, this network that is very involved in your awareness of the environment disconnected. So it was very exciting. So once we knew that, we could validate this study, this method to study diseases, and I'm not going to go over details, but we have done here just listed 2 studies, one we did in Tourette patients, another in Parkinson's disease, where we can see changes in connectivity between group of patients, and we and others have done this in many, many disorders. And we try to understand what are the brain networks that are affected. So now I'm going to tell you about a very different project, but also we try to understand networks and how the brain connects to each other from a different viewpoint. And this is on deep brain stimulation. This is a project starting a completely different angle. NINDS have a DBS program where it helps to train physicians on learning how to treat these patients and program. The stimulators is a big treatment out in the clinics. There are many cases, even though it's not fully understood. But within the process of the clinical treatment, the neurosurgeons need to have a set of images to be able to do their planning. So, and then the neurologists have a set of clinical information to decide on how to program these stimulators. So we have all this information available, and we could do something with that, right? So this is what we did. We got myself to be the cover girl with a MRI selfie. So this is the best we can do. So now, seriously, we decided to look at all that data, and as we start trying to register these images, we have MRIs with different contrast, T1, T2 diffusion. Diffusion is the way that, I mean, you probably have seen in magazines or some other places these little coal lines around, and what they do is try to decode the image collected in different directions to decompose and find out what is the predominant direction of propagation of the water. This is a technique that was created here at NIH as well many years ago, and is very widespread use. And you can see where things go and how things connect in the brain.
Silvina, yeah. Can you say just something short about the physical, how it's done, putting the electrode in?
Yes.
In case they don't understand.
Yes. So yeah, the surgery. Let me go back to my selfie. So the way that surgery is done, they get all these images and they choose a target. In this case, is the subthalamic nucleus. That is a deep structure in the brain. Sometimes for Parkinson's, it's also done in the globus pallidus. So they use these images to navigate where they are going to put the electrodes. Then they will do a hole in the brain, pass electrode. I mean, this is a cartoon because we didn't want to put, for privacy issues, a real patient. But the real data will look kind of like this. There is a coil, like a set of cables, and there will be two contacts placed inside the head that they will measure the angle and the distance on where to put. There is a stereotactic frame attached to the head of the subject, so they can know exactly where they are. And also during the surgery, as a neurosurgeon is putting the contact in the electrode, that electrode records the brain signal. So there is a monitor, and different neurons have different patterns of firing. And so they can, as they are putting the electrode in, they can monitor and find the sound or the shape of the subthalamic nucleus for cells and decide that they are in the right position. But then, of course, there is some imaging post-op to verify that you are in the right position. So, yeah, any of the things, everything that I'm telling you, probably we can spend an hour or more talking about. So I just wanted to give you a brief idea. So I'm trying to rush through this because I'm getting short on time. So we tried to find out where we are, and so this is a real contact. This is data from a real patient where we automatically can detect where this electrode is, taking this out, peeling off the CAT scan to the level that we get the metal only, and then knowing the geometry of the contacts, we can decide where these blue dots are, that the part that would send the stimulation, and then knowing the programming, the clinical part, what values were used for simulating the patient, we can simulate these spheres. So what we can then do, getting all these together after processing the data, and I'm happy to talk to you later, you can look at that website that I mentioned to see how these things are processed. We can put this sphere in here and just leave the parts that pass through it. And then we can study where this stimulator is going in the brain. We can start exploring the data. I mentioned to some of you early on that I will show a diagram. So this is what goes behind getting that little sphere in the right place in the brain. So there is a lot of programming that goes behind being able to do these type of studies. So don't forget the technology behind what you're doing. And we can then look at different places in the brain, and this is the same person, the same brain, but different locations. And when I'm talking about these locations, I'm talking about millimeters away. We can look at where these things go and start trying to make sense. I should say that the first author of this, Pete Laurel, was an ARTA in our program, in our lab, so they're doing great work. We can look at these. I'm going to flash through these so we get more lesson time. We can see where the more relevant thing goes, where every patient that have a good clinical outcome get the connection. And what this will allow us is to be able to do more predictions and try to eventually get to see the side effects. So can we avoid side effects? So a couple of slides, and I will be done in a few minutes late. Some of the, using neuroimaging, we can try to understand how the brain works. We sometimes have things that work, like for example, DBS. We know that it works, but we don't know how. So using the neuroimaging, we may start gaining some insights, and neuroimaging and of course electrophysiology, some insights into the mechanisms, and by one hand, make it more efficient, both in the treatments and in the decision of who could benefit, and look at alternatives for treatment as well, understanding how you can compensate the system. And one of the big problems when you try to, in general, correlate all these neuroimages or imaging studies with clinics is that sometimes we define a disorder in the clinic does not have a clear biomarker in a quantitative, being in the images or in the cell cultures or in the blood. It's very hard to find the biomarkers. The biomarker needs to be something unique to that disease. It needs to be that if you have the thing, you have the disorder. And even genetics, you can have a gene that is a carrier for a disorder and doesn't mean that you will become affected. So it's a very hard thing to do, to try to understand when something will become a disorder or a problem. And we are trying to use neuroimaging and techniques from a lot of engineering bases to understand this. So I will say a couple of words on this. As I mentioned before and Rita said, my background and grad school are both in engineering. And I have experience being a woman in engineering for a long time. And I think that probably is the same for other minorities. And sometimes it's a challenge. I have had experiences where been talking about my work with a male colleague next to me, and the person that I'm talking to would address my colleague or not me. I've seen the same thing. That was in Argentina 20-something years ago. I've seen the same situation 2 weeks ago here in the US, in interaction of young female engineer students and senior people and their colleagues. And I assume this is the same in other minorities, and also been told that in other places, like in Germany, that you wouldn't expect, also is a similar thing. So for those of you that are the females or the minorities, be strong, don't get discouraged for the things, and make your point. For those of you that are on the other side, be aware and try to not be biased against these people. The same thing happened when you negotiate a job or a salary, things like that. Be, as Jordan was saying, trust yourself, value yourself, right? It's common to think that you have less of a chance to get a position because you come from a minority and they would expect someone else. And sometimes you would be willing to accept less because you come from there, but sometimes these come on us, right? We are not asking the same because we think that we are already given the chance to do it, so we can push the limit. You can push the limit. You should be valued as much as everybody else. So if you don't know what is the framework, ask your colleagues, ask your mentors, and try to value yourself and ask for it. So as I said before, I chose the title as of the chance. For many years, I consider myself very lucky for being at the right place at the right time. But over the years, also learned that you need to help that, right? Things don't happen by accident. All of you are here because you want to succeed, you want to build a base for your careers. And so keep going, be prepared, be connected, and be open-minded to positions. Don't block yourself in what you can do. Thank you.
Any questions? Can I ask you,
Yeah.
you were a teaching assistant for what course?
I was 8 years as a teaching assistant for the biomedical engineering lab.
So do you think that if these guys had an opportunity to be a teaching assistant, do you think that's important?
I think it's great, yeah. My experience, I mean, I miss teaching, right? I've been, actually more than that, I've been teaching assistant a few years in Argentina and then 8 years here in the US. And I think teaching is a great experience. You learn a lot. You need to anticipate the problems, right? And the first year you teach a class is definitely a lot of work, whether you're a teaching assistant or an instructor. Over the years, you realize the problems are all the same, and the third year around, you just don't even need to think much about it. But it's a great experience. You get to think about what's going on. You get asked questions that you may not have asked yourself, and you need to be quick, and you learn from the questions of your students or from the mistakes that they do. So it's a great experience. Definitely, I would recommend it.
A question in the back.
Yes?
That's awesome. I mean, it seems it's really fascinating, the whole idea of functional MRI. But where would you really put the technology right now, and where do you see it like in the next, 5, 10 years? I'm thinking, I don't know if you've ever seen that movie Chappie, where you can actually see consciousness on a computer, or maybe even a functional MRI. And where do you kind of see that in the next 10, 20 years?
Well, I think to see consciousness, first we need to understand consciousness, and that's different people will have different opinions. I think it's one of the very interesting questions, and I wish we could understand consciousness, in particular, not that much on the science fiction level, but really on patients with disorders of consciousness, coma, vegetative states, and try to understand what we can predict for them. And I think in that field, hopefully, in 20 years, we would be ahead of where we are. In terms of brain decoding, we are moving towards certain targeting. It's a very hype in the field to try to decode things. So what they call, so there are certain patterns and templates that today you can get on that. What we are moving is to have stronger magnetic fields, higher resolution, more precision. We still need a lot of integration. There are huge initiatives to try to bridge the gaps between the high, like macro level that we have now and a lower level, and we may get more into that. I think still some of the things are slow pace changing, but we're making progress.